I was to give you a tall pea plant and a short pea plant, and just by looking at the phenotype, you had to tell me the genotype. Well, it would be very easy in the case of a short pea plant. A short height represents a recessive character. Recessive character is only expressed when both the alleles are in a homozygous condition. एक भी heterozygous आ गया, dominant allele आ गया, the recessive trait is not going to be expressed. So while looking at the short plant, you quickly tell me that the genotype is going to be both recessive. But what about a tall plant? Just by looking at it, would you be able to tell me the genotype? Well, two cases are possible. Remember, one is the homozygous dominant condition, other will be a heterozygous dominant condition. So how are you going to tell me the genotype? Hello guys, welcome back to Learning with Isha. Today we are looking for the solution of this problem, and the solution to this is a cross named as the testcross. The name test itself represents that we are testing the genotype of a given organism. How do we do that? Well, what we will do is we are going to cross this plant with a recessive parent. So the unknown is crossed with a recessive parent, and whatever outcome is going to come is going to indicate the genotype of the organism. Let's quickly begin and start doing that. Let's quickly look at the two cases possible. The first case is where the unknown organism is homozygous dominant. So the genotype is going to be, we are taking the height character as I have already told you, and we are going to cross it with a recessive parent. Okay, so this is my unknown and this is my parent. Well, the next step is going to be the gamete formation. The gamete is going to be, by this time we are all very fluent in writing all these steps. Now the next step is going to be what? To perform a fertilization. For that, let's quickly draw a Punnett square. So we've drawn the Punnett square now and quickly listed down the gametes. Now this is for my unknown and this is for my parent. Well, let's quickly now fill in the boxes as well. So what we observe in the case of a homozygous dominant parent is all the four offsprings are of the heterozygous type. So these heterozygous offsprings represent what? They're all tall in height. So they're all tall. So the genotypic ratio in this case is going to be 4:0. This is my genotypic ratio. My phenotypic ratio is also going to be 4:0. 4 represents that all the four offsprings are tall. 0 represents that there are no offsprings of the other type. Means there are no short-heighted offsprings. Now let's quickly move on and repeat the same process for the heterozygous parent. The genotype in this case is going to be of the heterozygous type. Again, we are going to choose a recessive parent. Quickly, we are going to form the gametes. In this case, the gametes will be of the same type. Quickly, we are going to again go back and draw the Punnett square. Quickly listing down the gametes in each case. Filling in the boxes now. Yes. So what we obtain in this case is that the offsprings are all not of the same type. Two types of offsprings are being seen. One of the heterozygous tall type and other of the short height. Well, so the genotypic ratio in this case is going to be what? The genotypic ratio is going to be 2:2, as in 2 heterozygous tall plants and 2 short-heighted plants. So this represents a 1:1 genotypic ratio. Well, the phenotypic ratio is also going to be tall is to short. Tall plants are these two. So 2, and short plants are the recessive ones, again 2. It's again going to be a 1:1 ratio. So we see that the ratios in both the case are different. Let's quickly conclude the results for the test cross for the monohybrid cross. This was the monohybrid cross. What we obtained that when the unknown was of a homozygous dominant type, the genotypic ratio and the phenotypic ratio both were 4:0. This means that जो भी offsprings होंगे सब एक ही तरह का character express करेंगे, वो होगा dominant type का. The second case was when we had the unknown of a heterozygous dominant type. What we did was we quickly crossed it with a recessive parent. What we got was a 1:1 genotypic and phenotypic ratio. इसका क्या implication है that the offsprings, half of them are going to show the dominant phenotype, but the other half are also going to show the recessive phenotype. How can we go backwards? Now, if what I have is that the result is that half of my offsprings are of recessive and half of the dominant, I can work backwards and quickly tell that my unknown organism must be of a heterozygous dominant type. A logical question that might come to your mind here is कि हम ये homozygous condition के लिए recessive में क्यों नहीं test करते? Like, why could the unknown organism not be of this type? Well, the answer to this is कि recessive condition में testing की ज़रूरत ही नहीं है. The short height is only going to be expressed in a homozygous recessive condition. So, no testing in this case is required. Now, in the next video, we are going to talk about back cross. Thanks for watching the video. Please consider subscribing to the channel to be updated about more such videos. Thank you.